Hi, everyone. I'm happy to be here remotely. Sorry I couldn't be there in person. I'm presenting today on a large project by a large group of people, as you can see in our list here. We're focusing today on the lunar analog spectra and trying to understand changes in lunar hydration that were observed recently by our team. And you can see in these images here, for the morning, we have on the moon, we see more hydration, which is mapped in blue. And at midday, we see less hydration. So in the Mare regions, we see more hydration in the morning and less hydration at midday. And we see variations from Mare to highlands across the moon. And so that's what we want to try to understand through our analog study. So if I could have the next slide. M-Cubed, the Moon Mineralogy Mapper, is mapping the surface of the moon, or did map the surface of the moon with spectra from about 0.4 to 3 microns. And you can see here the spectra with mineral bands that we've been talking about, as well as the hydration signature. And I realized I just didn't put my camera on, so there we go. The hydration is observed in this region near 3 microns. Unfortunately, M-Cube cuts off right at 3, so we can see a snippet of this hydration band due to water and OH. And so we're using an integrated band depth called IBD in order to map a portion of this across the moon. Next time step, please. So a number of studies have analyzed M-Cube data, including characterizing this 3-micron hydration band, thermal corrections, variations in the hydration with composition and location across the moon, and variations in hydration with time. And I'm not going to focus on those today, so let's go to the next time step. So how do we compare, where our goal is to compare relationships between the lunar hydration and the surface composition. So how do we go about this? First, the M-Cube data needs to be calibrated, and Christian Voller and Kai Wolfarth have done that for our group. And then generate these hydration maps, which Kai Wolfarth did for his PhD thesis very recently. And then the next step is to resample lab spectra of lunar analogs and potential minerals in order to characterize what the shape of this hydration band and other mineral features look like on the moon. And then we've been resampling the spectra for targeted and global channels and then comparing these to the lunar data. So the next time step. The next slide. There we go. Last summer at the SETI Institute, we had a Frontiers Development Lab focused on the hydration on the moon, and these are 5 of the young people who worked on this with Mario Parente and I, and they were using machine learning to evaluate the M-Cube data with a focus on the hydration. Next slide, please. So looking at the moon, again, on the left, we have a map of this integrated band depth with increasing blue in the Mare regions and some other places due to increasing hydration. And then on the right is an example of a map of pyroxene with increasing intensity in red. And this is sort of what motivated our study, where we see increasing hydration, where we see increasing pyroxene present. And that's something that Kai is focusing on investigating. And the next slide, please. So in order to get a collection of lab spectra, we first went and looked at what had been done, and we found that Roger Clark had processed his spectral library at the USGS for the M-Cube targeted channels, and this means 10 nanometer sampling across the spectral range of M-Cubed from 0.446 to 2.991. And Roger did this for his for his sets of 2 data, and shown in blue and red here. And so we wrote some code to do this as well for ours, and we compared it with Roger's data and confirmed that it's working properly. And one thing you can see on the right, for 3 different types of pyroxene, there are changes in the spectral region near 3 microns, which is what we wanted to evaluate. So the next time step, please. Then we used our code to process a wide range of spectra from the Relab database at Brown University, and also a few more of the USGS data that had been spliced together. So we have one spectrum across the whole M-cubed region. And so on the left are some volcanic glasses, and then some feldspars, and then a bunch of pyroxenes and some olivines. So next time step, please. And then we wanted to convert these targeted spectra also to convolve them for the global channels on in the M-cubed database, because most of the data collected by M-cubed is in this global mode. Only a few targeted channels were collected. And so converting the targeted data to the global data is a little bit more tricky. They bind the data at 4 data points, but then the steps change from 2 to 3 to 4 to 5, depending on where you are in the spectrum. And this was done in order to get more data near 1 micron to characterize differences in the different kinds of ion-bearing silicates. So we wrote some code to do this and tested it on a number of different samples. And so as far as we know, this isn't available anywhere. So we're preparing these for a number of lunar analogs so that the community can use these as well for analyzing M-cubed data. Next time step, please. So going back to the M-cubed data then, we wanted to compare spectra from 5 locations and with data from our study from the lunar analogs. And so these 1, 2, 3, 4, 5 spots here collected 2 from the Mare region and 3 from the highlands. And on the bottom, you can see the individual spectra in blue from one highland spot, and in green from one Mare spot, and then ratioed spectra at the top, so ratioed from morning to midday. And so we're mostly focusing on a change in the hydration band from morning to midday. And then you can see there's a difference in shape here for the blue and the green, indicating a difference in the hydration. So let's go to the next time step. Here again are those 5 locations, 2 and 4 from the Mare, and 1, 3, 5 are from the highlands. And if we look at the green Mare spectra ratios, they tend to be increasing past the end of the M-cubed channels. So the hydration band, the change in the hydration is increasing. Whereas for the blue highlands type, there seems to be more of a bowl type shape or it either plateaus or goes back up, indicating a different shape in that hydration feature. And so we compared these with a bunch of lab spectra as well. Next slide. And so this is zoomed in more on the hydration region. So we think this indicates there are at least 2 forms of hydration present on the moon, which is really exciting. And probably a number of people suspected this already because of the variations across the surface. But what we see here are different shapes in the kinds of hydration for the highlands and the Mare, which is oversimplified, and we're going to be looking in more detail at certain regions. But for now, I think it's interesting to look at the highlands versus the Mare to characterize these 2 different regions. And if we compare these with lab spectra, we also see a difference where some pyroxenes, including augite, have a greater intensity near 2.8 or so, and other minerals are increasing at longer wavelengths.
1 minute.
Next slide. Okay. In summary, then, we resampled a bunch of lab spectra for comparison with M-cubed spectra. And first we convolved these to targeted channels at 10 nanometer steps, and then to global channels at 20 to 40 nanometer steps. And we observed persistent hydration across the highlands regions. And this includes a spectral change from morning to midday with a bowl-shaped hydration band or plateauing band. And the changes are more similar to augite or pigeonite, 2 types of pyroxene. And then elevated hydration bands are observed in the Mare regions in the lunar morning. So the Mare have more in the morning and less at midday. So it's increasing change. And increasing shape in the hydration band at wavelengths past what M cube can observe. And so this shape of this is more similar to anorthite and many of the other analogs we investigated. So thank you for your attention today.